Diversity of marine phototrophs. Introduction to marine phototrophs. In organic compounds, phototrophs transform the energy of solar irradiation into chemical bond energy. These organic substances provide biological energy not only for the activities of life on Earth, but also for regional and/or global ecosystem formation and maintenance. However, the developments of technologies for phytoplankton research within the past 3 decades reveal a diversity of phototrophs serving as primary producers and contributing to the transformation of light energy into chemical energy. Diatoms and flagellates are known to be the largest ocean producers. Traditional understanding of primary producers. It was acknowledged that primary development in marine environment was mainly carried out by diatoms and flagellates. In a broad variety of water quality, including eutrophic and oligotrophic environments, phytoplanktons are distributed. Dinoflagellates are also widely distributed in the oligotrophic ocean as a key member of phytoplankton. From the approach to phytoplankton sampling and the methods for phytoplankton detection at the time, the explanation why those two macroalgae were considered to be the main primary producers was easily speculated. Plankton nets had been used for phytoplankton sampling. A plankton net is field equipment used to track plankton. In marine environments, the dominant phytoplankton has a much smaller cell size than the cells that the smaller mesh-sized nets can capture. Research on phytoplankton using deoxyribonucleic acid, or DNA, detection and flow cytometry has shown that in marine habitats, the predominant phytoplankton is much smaller than those collected by plankton nets. For example, haptophytes including Phototrophs, which are phytoplankton that deposit calcareous plates called coccoliths, are one of the major constituents of primary producers in marine environments. Recognition of Picocyanobacteria dominance. Cyanobacteria unicellular Synechococcus. They are mainly distributed in marine environments, especially in tropical and subtropical regions. They conduct using divinyl chlorophyll A equals to B oxygen photosynthesis that can effectively absorb the ocean-dominant blue light. They are the dominant phytoplankton between the latitudes of 30 north and 30 south in tropical and subtropical regions. In addition to Picocyanobacteria, known as Synechococcus, divinyl chlorophyll contain divinyl chlorophyll which had a significantly different absorption range from that of the normal chlorophyll A. Discovery of ubiquitous photoheterotrophs. Two important results were published in the early 2000s. The wide distribution of anaerobic anoxygenic photosynthetic bacteria AAPB, which contain bacteriochlorophyll A. They had mainly been recognized as a photosynthetic pigment in anaerobic anoxygenic photosynthetic bacteria (NAPB) distributed in anaerobic environments. The wide distribution of proteorhodopsin-containing bacteria. NAPB was understood to be distributed without oxygen production in an anaerobic environment and to perform photosynthesis. AAPB grow heterotrophically in an aerobic environment. The rhodopsin-like protein that is found in marine proteobacteria is proteorhodopsin. It is a complex of retinyl opsin in an rhodopsin member. Opsin is a set of 7 transmembrane bundles. Rhodopsin is well known as a photoreceptor in vertebrates as a light sensor. Proteorhodopsin has been widely and significantly distributed in the marine environment and has played an important role in the transformation of light energy. Both bacteria are photoheterotrophs without carbon fixation and had never been recognized as primary producers. Oxygenic photosynthesis using far-red light. Chlorophyll and is crucial for oxygen photosynthesis, as it plays an important role in the primary reaction of photosynthesis in light harvesting, energy transfer, and charge separation. Photosynthetically active radiation (PAR) is classified as the in-situ light absorption range of chlorophyll A from 400 to 700 Nm or from 380 to 710 Nm for the evaluation of light intensity for oxygen photosynthesis activity. PAR is used. Chlorophyll D is a red-shifted chlorophyll which can absorb far-red light for an almost 30 Nm longer wavelength than chlorophyll A. The efficiency of the energy transfer of photosynthesis using chlorophyll D is equal to that of chlorophyll A. Chlorophyll F has its absorption maximum at around 720 Nm and it can absorb far-red light up to around 760 Nm in vivo. Discovery of eukaryotic phytoplankton. Picohaptophytes make up 5% of global biomass of phytoplankton and provide an essential element to primary production in the marine world. This showed that a significant amount of picohaptophytes, including many undescribed species, were widely distributed in marine environment. Several studies on marine phytoplankton research using the metagenome and flow cytometry techniques were published around 2010. By metagenomic analysis of a plankton fraction of less than 3 micrometer in size that was tentatively called picobellyphytes. Phytoplankton which did not belong to any known phyla of algae was identified. Picobellyphytes distribute widely in coastal areas of Europe and the North Atlantic. Metagenomic analysis targeting plastic DNA, which was tentatively called Rhapemonads, also detected the new candidate phylum in 2011. Rhapemonads widely distribute in coastal areas of Europe and the Atlantic and Pacific Oceans. Strange phototrophic microorganisms. Through metagenomic study of nitrogen-fixing bacteria in marine waters, the cyanobacterium-like particles UCY was found. The broad distribution of UCY and A has shown that the particles in pelagic waters contribute to the nitrogen cycle. UCY and A lacks photosystem II, one of the two photosystems that split water into protons, electrons, and molecular oxygen. Rubisco, ribulose 1,5-biphosphate carboxylase oxygenase, the primary enzyme for fixing carbon dioxide and the tricarbolic acid (TCA) cycle for respiration. The lacking of these essential genes for free living suggests that UCY and A might be a symbiont in some organisms. Diversity of light energy transformation systems and reconsideration of photosynthesis. Organisms that can grow using light as its energy source are called phototrophs. Representatives of phototrophs are photosynthetic organisms that can perform photosynthesis. Photosynthesis is an action that can reduce carbon dioxide into organic compounds such as sugars using sunlight as an energy source. Primary producers are species that use inorganic carbons as their carbon source to synthesize organic compounds. This is because of the productivity of chemoautotrophs in a general environment is considered to be much smaller than that of photoautotrophs. Primary producers has been used synonymously with photosynthetic organisms. Phototrophs are species that can use the transmembrane electrochemical potential of proton to synthesize ATP by ATP synthase by pumping protons from one side of the membrane to another side based on light energy. Protons are pumped out of the cytoplasmic space into the preplasmic space for non-oxygenic photosynthetic bacteria or proteorhodopsin-containing bacteria. Protons pumped into the thylakoid lumen or pumped out by ATP synthase from the cytoplasm flow back into the stroma or cytoplasm, respectively, which results in ATP synthesis. Here, all species which can perform such light-dependent ATP synthesis are phototrophs. Traditional versus new understanding in primary producers. 1970. The atoms and flagellates are primary producers. New species has been found unicellularly: Cyanobacteria synechococcus. 1980. Chlorophyll as a performs oxygenic photosynthesis to primary producers. 2000. AAPB and NAPB were the two bacteria primary producers that had been reported. 2010. Pico-eukaryotic phytoplankton had been discovered. 2011. Strange phototrophic microorganisms had been discovered. Conclusion. Energy flow studies are significant and applicable to global environmental change, the sustainability and conservation of living marine resources, and the diversity of marine microbes in relation to the food web in marine environments. The diversity of marine phototrophs and their function in energy and material flow in marine environments need to be exposed in order to transition to a new paradigm and to establish a new understanding of energy flow in marine environment.